Welcome everyone. Here in the previous classes we have seen that is what are the different conditions required for the growth, what is growth, as well as growth rates, that is relative growth rate, that is absolute. We have seen this different growth rates, arithmetic growth rate and geometric growth rate. We have even seen absolute growth and relative growth. Here now we will move to another topic, that is differentiation, re-differentiation and de-differentiation. We know what is differentiation. We have already studied this. Cells after they divide, they grow. After the division, the cells grow, increase in size, they elongate. Then after elongation, now the cells will differentiate. Differentiate in the sense the cells will be assigned a particular task. The cell will be given a particular function to perform. That is, the cells will be given task or function to perform. For example, the cell after the formation of zygote, here zygote will be continuously dividing and it produces new cells, many number of cells, a mass of cells is produced. After the formation of zygote, when the zygote starts dividing by mitotic division, zygote produces a mass of new cells. Those new cells, for example, if there are 100 cells, so out of this 100 cells now, here 10 will be given function, 10 cells will be given function to transport water. That is, they will be forming xylem. 10 cells will be given function to transport food material. That is, they will be forming phloem. 10 cells will be given function to protect. That is, they will be forming epidermis. So 10 cells will be forming ground tissue, vascular ground tissue, or some parenchymatous cells, different. The cells will be assigned. First, a mass of cells will be formed, the cells will grow and they will be assigned a particular task. After the growth of the cell will be given a particular function that you have to perform a particular task. You have to, for example, 10 cells are given task of transporting water, that is, they will be forming xylem and through that is, the function of them is to transport water. When the function is given to them, that is transport of water, those cells will only help in transport of water. They does not perform any other function. They are given a particular special function, they will be performing that particular function only. They won't perform other functions. For example, if a cell is given function to transport water, it will be only transporting water. It will not transport food. It will not help in protection. It will not help in, that is, giving the strength to the plant, it will not help in other functions. It will perform only that function which is given to them. So this process of giving a particular task to the cells, that is called as differentiation. The cell when it grows now, two major changes will be seen in the structure. After growth of the cell, after division, the cell will be growing and there will be many structural changes which takes place. One, that is, two of the major structural changes which takes place is here protoplasm concentration and cell wall. For example, if the cell is given function to transport water, then what happens? The protoplasm, here xylem will be having, xylem tissue is made up of many different cells, that is, tracheids will be there, vessels will be there, parenchyma will be there. If we take an example of tracheid, Tracheid, it is a type of cell present in xylem tissue. Here this will be helping in transporting of water. So it is almost a dead cell. Here inside the cell, before undergoing changes, protoplasm was this much and the cell wall was very thin. Cell wall was very thin. After assigning of a function, what happens is the cell loses protoplasm. Only little protoplasm will be present, and around the cell a very thick lignified wall will be formed. Very thick lignified wall will be formed. That lignified wall will not allow the water to come out, as well as it gives strength to the cell, and the cell has to lose its protoplasm. In order to form a tracheate, the cell has to lose its protoplasm. Earlier, before assigning a function, protoplasm was in more amount. But if it is forming a tracheate, then the cell has to lose protoplasm, and the cell should have a deposition of lignin on the cell wall. So it gives the strength. See here, two structural changes will take place. Many will take place, but the two major changes is cell wall, that is deposition of lignin, pectin and all those impervious substances will be seen, and protoplasm content increases or decreases or remains same depending on the type of function they will be performing. Here in the xylem, apart from tracheate, fibers will be there. Fibers, parenchyma. In xylem tissue, we find xylem parenchyma, we find fibers, we find tracheates. Tracheates and fibers, in both the cases, the cell will lose protoplasm and lignified wall will be formed. Whereas in case of parenchyma, here the cell will not lose protoplasm. If it is forming parenchymatous cell, if the cell is becoming parenchymatous, then it will not lose the protoplasm. The protoplasm will be as it is. But if it is changing into a fiber or if it is changing into a tracheate, then it loses its protoplasm and the cell wall will become thick with lignified thickenings. So this changes takes place. Here when the cell grows, it undergoes structural changes, and after undergoing structural changes, the cell matures and they will be performing a particular task. This process of assigning a particular task to a cell, that process is called as differentiation. The process of assigning a particular task or a particular function, that is the process of giving a particular function to a cell to perform. So that process is called as differentiation. That is giving work, special work to the cell. Now after a particular function is given, after a particular work is given, it will be carrying out that particular function only. It won't carry out any other function. It won't carry out any other function. Now coming to this, another process, that is dedifferentiation. Another process, that is dedifferentiation. Here when the cells differentiate, they lose their ability to divide. One important thing here is, after the cell is given a particular function, they will be carrying out that particular function only throughout their life, and they will lose their ability to divide. After cell is differentiated, they will lose their ability to divide. It will lose ability to divide and matures. The cell will lose ability to divide and matures. So that happens in differentiation. But another process, that is dedifferentiation. In this what happens? The differentiated cell which loses its ability to divide, it will again change into, it will again regain their ability to divide. Here, after differentiation, the cell has lost its ability to divide. But in few cases, few cells, even after losing their ability to divide, they will again regain their ability to divide. For example, here cells of interfascicular cambium. We have studied this in anatomy in first year. In anatomy, here this vascular tissue, this is this. This is phloem, this is xylem, and here this is phloem. Here inside this we have seen, we know this one, inside, in between this xylem and phloem we have interfascicular cambium. This one is called as interfascicular cambium. Interfascicular cambium, and here parenchymatous cells will be present. In between these bundles, parenchymatous cells will be present. These parenchymatous cells have been given a particular task, that is parenchyma. They have been differentiated into parenchyma. They will be carrying out a particular function. Parenchyma will be helping in different functions to serve, that is to store food material, to help, it depends on the location, and depending upon the location the parenchyma will be performing various functions. So now parenchyma are the cells which have been differentiated. They have been given a particular function, that is to store the food material. For example, to store the food material, here what happens, they will be providing nutrients to the cells. They will be providing nutrients to the cell. Now these parenchyma, what happens to this? Now they doesn't have the ability to divide. Now parenchyma, when they have differentiated, they lose their ability to divide. But sometimes what happens is, this parenchyma, few of the parenchymatous cells, not all, few of the parenchymatous cells which has lost their ability to divide, they will again become meristematic. They will again become meristematic. Here parenchyma, the cells has been differentiated and changed into parenchyma, that is they have lost their ability to divide. But under certain conditions, this parenchyma will again change into meristematic cell. Not all parenchyma, few of the parenchymatous cells will change into meristematic. That is, if they are meristematic means then they will have the ability to divide. Here they lost ability to divide. The cells after differentiation, the cells after differentiation has changed into parenchyma. Now this parenchyma has lost the ability to divide. But under certain conditions, few of the parenchymatous cells will again change into meristematic tissues. Meristematic tissues, they will have the ability to divide. Here they lost the ability. After changing into meristematic cells, they again regain their ability to divide. Now this process is called as dedifferentiation. D means here completing deletion of differentiation, that is removing that particular function and again changing into meristematic. These meristematic cells, when they grow, they differentiated into parenchyma, lost their ability to divide. But under certain conditions, these parenchymatous cells again changed into meristematic. Not all, few of them. Here many are there, only few of them has again changed into meristematic. Again changed into meristematic cells, and they have gained their ability to divide. So whatever function was given to them, that has been deleted. Now they are again meristematic. They are again meristematic. So this process where the differentiated cells which has lost their ability to divide, under certain conditions regain capacity to divide again. So this process is called as dedifferentiation. This dedifferentiation, this is an example of dedifferentiation. The cells in between the xylem and phloem, that is intrafascicular cambium. This one is called as intrafascicular cambium. Intrafascicular cambium. So this is an example of dedifferentiation. The parenchymatous cell which has lost their ability to divide, under certain conditions they will again change into meristematic. They will again change into meristematic, and they have their ability to regain their ability to divide. That process is called as dedifferentiation. Same is seen in cork cambium also. Same is seen in cork cambium. Another example of this, like this intrafascicular cambium, here another example is also cork cambium. Now coming to this, another, that is re-differentiation. Re means again. Re means again. Then what happens? They have ability to divide. These cells which has lost their ability to divide, they have changed into meristematic. They have gained their ability to divide again. So then they will divide again, and after dividing they will lose their ability to divide again after given a particular function. When they have changed into meristematic, they won't be meristematic throughout their life. After changing into meristematic, that is cells, they will again divide, they will regain their ability to divide, they will divide, produce few cells, they will grow, and again they will differentiate and perform a particular function. So new cells will be formed in order to perform a particular function only. So new cells have been made by the activity of the meristematic cells, and after new cells are formed, they grow, and then again they get differentiated. So this process is seen in dicot stems and dicot roots, where we see secondary growth. So the process of assigning a particular function to a cell, after which the cell loses their ability to divide, that process is called as differentiation. Then that the differentiated cells which has lost their ability to divide will again regain their ability to divide under certain conditions. That process is called as dedifferentiation. And these cells which regain, the newly regained cells which has gained the ability to divide, they will again divide, and then they will get again differentiated. So that process is called as re-differentiation. Be clear in this. So we have seen what is differentiation, that is assigning a particular task to or special function for a cell to perform. In the next class, we will see what are growth regulators.